In this video, I'm going to be talking about proteins, and we're going to be talking a lot about protein structure, which protein is obviously a source of nutrients, but its proteins are also the molecules that do all of the metabolic work in our bodies during metabolism. And so talking a bit about protein structure is going to aid both in, well, mostly in thinking about proteins as the workhorses in metabolism. It's not going to be as much about protein insofar as it is nutrients, because when we think about protein as nutrition, we're usually thinking about the amino acids that compose the protein. So when we eat protein, our body actually has enzymes that break down the protein into the amino acid constituents, and that's where that becomes nutrition. So this will be mostly about protein structure. And so to get into it, the first thing I'm going to point out is when we talk about protein, we talk about it in, we first want to define sort of what the size of a protein is. And so that's what this table over here is showing, is that we can define the size of protein by the various sizes of the amino acids that are contained in the protein. So this is the list of all the amino acids with their 3-letter and 1-letter abbreviations. But then we go over here to the molecular weight, which with proteins are usually done in Daltons, or actually more often when we're talking about proteins, because they're so large, they'll be in kilodaltons or even megadaltons. And so a Dalton is, well, it's defined so that 1 gram is equal to 6.02 times 10 to the 23 Daltons. And so what we can say is that 1 gram is equal to 1 Dalton, or no, so it'd be the other way around. It'd be 1 Dalton is equal to 1 gram per mole. Yeah, so uh, then obviously 1 kilodalton is equal to 1,000 Daltons. That's just your dimensional analysis. But this should be 1 Dalton is equal to 1 gram per mole. And the average amino acid, so the average amino acid is equal to about, should make this approximately equal to 110 Daltons. And so if you have, say, a protein that contains 100 amino acids, then you know that it's going to be approximately 1100 Daltons, which is about 11 kilodaltons, or uh, actually it would be 1.1 kilodaltons, wouldn't it? Yeah, because if we divide this by 1,000, which is our dimensional analysis, then we end up with 1.1 kilodaltons. And so that's how we talk about the size of proteins. And so a lot of proteins are going to be on the order of 50 to 300 kilodaltons. That seems to be sort of around the average, maybe even smaller, and obviously sometimes larger. For instance, uh, large protein uh, structures that contain multiple proteins can get up into the megadalton range. For instance, a protein that I worked on in my graduate work was in the megadalton range because uh, it was several dozen proteins uh, bound into a large complex. Uh, all right, so proteins, like I said, are composed of amino acids, and so we see here we can have multiple amino acids bound into this would be called an oligopeptide here, since it's more than two amino acids, but it's not enough that we would call it a polypeptide. But you could keep adding amino acids onto this to turn it into a polypeptide. What I want to show here, though, so see we have tyrosine, glycine, phenylalanine, leucine. What we want to see here, though, is on this end, we terminate at this primary amine here, where at this end we terminate at this carboxylic acid here. And so this is the amino terminal residue, usually called the N terminus, N because it's nitrogen, and then this will be called the C terminus because carboxylic acid C. So that's how we sort of define one end. So there is a directionality, as you can see here, to protein chains, and the directionality is defined as the way that amino acids are added onto the growing chain when we're translating into proteins. And so these are connected together by these peptide bonds, as I talked about in the previous video. And so this is the sort of the thermodynamics of peptide bond formation. So we start with two different amino acids. So we'll have our NH2 going to there, and we'll have our carboxylic acid that's free here going to some amino acid. And then to bind these together, so I'll call this R1 and R2, so we'll end up with R1N down to C like this, R2. And so this is the transition state where, like I said, that's that delta G with this fancy symbol. So back in the thermodynamic video, I talked about transition state, or we can think about it in terms of activation energy, which is the energy difference between the transition state up here. You can see it over here, but I'll redraw it over here. So it's this change in energy here, because we need to, if we have these two species, we need to add in enough energy to get all the way up this hill, and that will get us to the transition state. And then it goes back down here, and you see there's this delta G difference here between these two. And so that is going to be energy that we lose. And so building proteins out of amino acids actually requires energy. And if I'm not mistaken, I believe it takes, and we'll talk about these energy molecules more in a later video, but I believe it takes 4 ATP equivalents. And in a later video, I'll talk about why we talk about equivalents, because ATP is sort of the so I had a biochemistry professor say that it was kind of the $20 bill of energy in metabolism, because it's sort of the most used, and it's kind of, when we think about energy, ATP is the molecule that's sort of this is the $20 bill. That's the one that's often used, but there are other things that are used, things like GTP, which is basically the same, and that's why we call it equivalent. And 1 ATP, I think, gives uh, is when you use this as energy, it gives off about 30 kilojoules per mole. So if we have 4 here, then that will be 120 kilojoules per mole. And so that's how much energy we have to put in to make to bind 2 amino acids together. I won't dwell on that too much because we need to this. So this right here is what we call the primary structure of protein, and that's just the sequence of amino acids. But then we want to go off into what's called secondary structure, tertiary structure, and quaternary structure. So secondary, which I'm actually going to talk about after tertiary structure. So tertiary structure is sort of the full 3D structure of the protein, and tertiary structure is made out of the sort of 3D orientations of these various secondary structures. And so, like I said, I'm going to actually talk about the tertiary structure first, and then I'm going to get into the secondary structure. And the reason for that is, I mean, obviously secondary structure is part of tertiary structure, but I think secondary structure can be sort of talked about in isolation, and tertiary structure is sort of a higher level thing that uh, makes more sense just in terms of sort of thermodynamic effects and things like that. So I'm going to talk about that first. And then quaternary structure, which is uh, the association of multiple different proteins together, like I was talking about. You can, that large multi-protein structure that I worked with in my graduate work is multiple proteins associated together into a large uh, complex. So they're called complexes. Uh, so I will talk a little bit about that, but uh, that I'll probably get into more once we actually start getting into the biochemistry and things like this become relevant. But I will mention later on sort of how that works. And so let's talk about this tertiary structure. So the first thing we want to talk about is how the tertiary structure actually comes about, and that is through what's called protein folding. And so when you are producing the protein, you have this chain here, and you're taking an amino acid and adding that onto the end of it. So there isn't any sort of obvious reason why this sort of unfolded protein is going to go down into the folded state, which is called the native state. Because especially since when we're up here, we're at this high entropy state where our protein can just sort of flap around in the breeze, so to speak. I mean, it's in solution, so it's flapping around in the water, but which seems like it'd be sort of the state that makes the most sense uh, because it's the higher entropy state. Then we can move down this entropy gradient to a lower energy, but also lower entropy, which you'd think would be less favored. But there are reasons that I'll get into in just a little bit why this happens. And one of the reasons, though, so first we'll talk about it this way. We can talk about how uh, we have this disordered protein. We go into what's called the molten globule state, which is sort of partially uh, disordered, but also partially ordered. And then through a stepwise sort of uh, process, this unfolded protein will sort of uh, go through various steps on the way down to this, and then it can go through various steps on the way down to this, and this is just some of the steps that we can go through where uh, these different parts of the protein are aligning into different spaces over in these different parts of the protein are forming. We get the loop formation and so forth, and finally we get to our native state. One of the things that does this is are these bond angles, or the phi-psi angles, the dihedral angles that I talked about, where we have our amino acid like this, and then we go here, then this goes up to there, and so this would go up to another thing like that, and so on. And we have our dihedral angles because this right here is going to be flat because of the resonance stabilization there, and this right here is going to be flat. And so we have our dihedral angles about this bond and about this bond here. And so because of different steric hindrances of like these carboxyl groups with the side chains, we have these allowed bond angles or dihedral angles. So dihedral, I don't remember if it's an A or so. I think it's spelled something like that, dihedral angles. And one of the driving processes of this this lowering in entropy is because we are in an aqueous environment. We are in solution of water, which, as we talked about before, is a polar molecule. So we have our partial positives on here and our partial negative up here, but our protein can have both polar side chains and non-polar side chains. So if we think of this as our our polypeptide, our chain that would look like this, and we have various different polar and non-polar side chains, then what's going to happen is in solution the polar side chains are going to want to face outward into the solution of water, because then we can form these hydrogen bonds and so forth with our side chains. So we can have our positive to negative or negative to positive hydrogen bonds. And so the hydrophobic side chains are going to want to go towards the interior. And this is what's called the hydrophobic effect. And right here I just have what's called a hydropathy plot, and that's just looking at the hydrophobicity of the amino acid. So this is the position of the amino acid in our chain. So we have our chain of amino acids here laid out in a straight line, so that this is the first at the N terminus of our protein, and over here is the last one at the C terminus. And this is just energy here. And so we know that if our protein, if an amino acid is in a favorable position, or if it's all right, to explain this more clearly, so here is our zero energy, and so it doesn't take any energy to be either in the hydrophobic or hydrophilic environment. So we see if we go down in energy here, we know that reduced energy is favorable. And so if this is looking at the energy of associating with the 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 aqueous environment, then we know that amino acids down here are going to want to be are going to be uh, hydrophilic, because these are it's going to be associated with water. It's going to have that favorable association with water. Whereas if we're in the positive energy, such as right here, then this is going to be hydrophobic. And so this is probably going to be on the interior of the protein. This is a particular protein called cytochrome oxidase B, and these right here are transmembrane regions. And so we'll get into this more once we get into biochemistry, but we know that the cell membrane, which we can just show like this, is is hydrophobic. And so if we have a protein that's embedded in this, then it's going to be something like this, where part of it is sticking out of the cell and part of it's sticking into the cell. So we're going to have this part of the protein here that's hydrophobic, such that it can be inside of the membrane. And so that's what these transmembrane regions are about here. But then we can look at other places here where we have positive energy, and we can pretty much be sure that that's going to be inside the protein, because these hydrophobic parts are going to want to repel away from the aqueous environment. All right, and so another aspect of this this hydrophobic environment on the inside is that there will be some of our protein or some of our amino acids inside that hydrophobic environment, and that is going to have an effect on the charge of the amino acid. And so what I have listed over here is the dielectric constants of different solutions. And we see here that water, which is our solution for proteins, has a dielectric constant of 78.4. Usually I've rounded that up to 80. And so what the dielectric constant is talking about is sort of how much charge is felt through the medium. And so the higher it is, the lower the charge is felt in the medium. And so basically what that means is, so if we have some atom over here that's positively charged, and maybe some atom over here that's negatively charged, if we're in water, then we know that we're going to have this sort of bonding like this with our species, with our ion here. So our partial negative is going to associate with this full positive charge, and over here it'll be the opposite, where our partial positive is associating with our full negative charge here. And what that's doing is stabilizing this charge. And so there isn't going to be as much charge felt between these two, since the solution is stabilizing this charge. And so having this higher dielectric constant is going to stabilize these charges such that they don't feel as much charge between each other. However, if we instead, I'll draw it over here, if we have our positive charge and our negative charge, and we're in something that's a nonpolar solvent, then there's nothing stabilizing these charges, and so there's going to be a lot more charge felt between these two, because our something like benzene, which is our ring structure like that. So if we have a bunch of those around, it's not stabilizing this charge as much. So this charge is sort of effectively stronger than what it would be when it's in a solution like this. And so we can look at the effect this has on the pKa, because as I talked about before, uh, stabilizing this charge is going to make it so that a charge like this, so if we had like an oxygen with a hydrogen on it, we are able to go to our oxygen, we'll call this the R over here, with a negative and an H with a positive. These are going to be more stable, and so this hydrogen is going to be more likely to come off, and so that's going to reduce the pKa. And so what we see over here is we're in this, this is from a paper where they made mutants of this protein, and so what they did was they changed some of these internal amino acids into this amino acid here, which is aspartate, and this has this negative charge on it, or it has this carboxylic acid on it, which can be, either in the protonated and non-protonated form, and that's going to depend on the pKa. So what they did is they changed a lot of these internal amino acids into, so E, as you can see here, is the one-letter code for aspartate. So they mutated these one by one to aspartate, and you can see here that the pKa is increasing. So we have these increasing pKa's, which means that this hydrogen is going to be less likely to come off. And what you can see here is, so in a nonpolar environment, in the inside of a protein, we have our pKa can increase with carboxylic acid. But with the amine group, then our pKa decreases, because in this case, releasing this hydrogen is going to give us a nonpolar right here, whereas here it's giving us a polar, or a charge here, where here we're getting a non-charged nonpolar. And so this is going to actually be favored, and that's why we're going to have our pKa go down, because we're going to be more likely to release this hydrogen, where here the pKa is going to go up because this is going to be non-favored. So this is what's called the Born effect. Then we can also, within our interior of the protein, we can have these charge-charge interactions. And so if inside this we have all sort of non-polar, but then say we have something here that's positive and something here that's negative, these two things, like I said, because we're in this non-polar medium, are going to basically have a higher effective charge. And so what's going to happen is, because these things can stabilize each other by together being neutral, we're going to reduce our pKa, because then we can have our charges stabilizing each other. And so this is another thing that can actually help maintain the shape of our protein, is that you can stick these sort of isolated charges into the interior and have them make this this ionic bond here, which can then further stabilize the shape of the protein. Which, just to sort of tell us what we're trying to do here, let's look back. We're looking at the protein folding and how, even though this is a lower entropy state, we can actually get to this lower entropy state. And the reason we can do that is because of this hydrophobic effect, which is going to give us this more stable very ordered form, and because of the hydrophobic effect, we can also get these ionic bonds, which are going to be stronger on the interior because of this hydrophobic environment. And so those are two ways that our protein can stabilize itself into this more ordered state here. And so looking back again at this uh, sort of an overview, we this is our native state. This figure calls it normal protein, but we can think of it as the native state, and that's this highly ordered, which, like I said, we're going down this well here of uh, of entropy. And just to uh, something I forgot to mention, this shows these little local minima here, and that would be sort of the steps taken to go down this. So this folded protein will get slightly folded here, and then it'll get more slightly folded, and it's sort of, it shows the molten globule here, but the molten globule will be kind of in this whole area, and then it won't be till it gets to this global minimum here that we're in our native state. And so what can happen is we can do things to this native state protein that can denature it, turn it back into this disordered state, disordered state. And so this is called denaturation, and there are different things you can do to denature the protein. You can change the pH. So we talked about up here things like these ionic bonds, and because we're in this hydrophobic environment, these charges will have different pKa's. But if we add enough H pluses or add enough OH minuses, changing the pH of our solution, then you will eventually change the charges on these. You might put hydrogens on here because you added enough of this, which would weaken this bond and allow this to start falling apart. And so changing the pH can change the structure of a protein, and different proteins are going to be in the native state at different pHs. And we'll get into more of this in the biochemistry. I said in a previous video that physiological pH is around 7.35, and one of the biggest reasons that you have to maintain this is so that we can maintain our native state of proteins. But there are proteins that can exist in their native state at different pHs. For instance, digestive enzymes can exist at pHs that are much lower, probably close to 1 or even 0. And so they would be in their native state at a much lower pH because they have to be in the acidic environment of our stomach. And so pH is one way that we can change that we can denature our protein. So temperature is another one. So I'll talk about that in just a second. Solubility is obviously one. If our protein is stable in a aqueous or water environment, so if we put it in, say, a environment of ethanol, then we would denature our protein because it has a different solubility in ethanol than it would in water, for instance. So temperature is another big one. And I show down here, this is actually data that I acquired in my undergraduate research looking at a specific enzyme called beta lactamase. So beta lactamase, it's a whole family of different enzymes. It's the enzymes that bacteria use to break down antibiotics. And so I was looking at different mutations. So this was what's called the wild type enzyme here. And then I was making these mutations in it, the serine to leucine at the 109th amino acid, in the glutamate to glutamine. Yeah, that's what D is. That's the glutamate to glutamine at the 222nd amino acid, the proline to serine at the 225th, and then putting the proline and glutamine or glutamate rather together into a single enzyme, and then putting all three of them together in this clinical variant called OXA 239. And so you can see just by adding these different mutations into our protein, we actually changed the stability of the protein, where you see that the OXA 23 with these two mutations denatures at a lower temperature than, say, the OXA 23 with just the serine to leucine, which denatured at this higher temperature. And so just by adding single amino acid mutations into our protein, we can actually change the stability of the protein in its native state. But you can see here that just by increasing the temperature, then our fraction of the protein in the folded or native state. So as we increase the temperature here, these stay in the native state right here. But then we start seeing degradation or denaturation of the protein starting here. So as we increase the temperature, we see quite a bit of denaturation just between these two temperatures, where something like this, like the wild type, which is in purple, and just the S109L mutation here, maintains stability to a higher temperature. And so we know that different amino acids have that effect on the stability of the protein. Yeah, so that's denaturation. And as this points out here, there's loss of biological activity. The structure of the protein is very important. In fact, it's everything when it comes to the protein's function. And so now we will start talking about these secondary structures. And first I'm going to talk about what's called the alpha helix. So alpha, that should be an alpha helix, which can come in two different forms. It can come in these left-handed helices and these right-handed helices. So you see here, they define this. So if you're pointing the thumb on your right hand up, then this curls in the direction of that your fingers are curled. Same with the left-handed. And when I talk about DNA, this will also come up. And so in our proteins, we see that we have our right-handed helix when our dihedral bonds are in this sort of area here. And when we talk about our beta sheets in a few minutes, those are up here. But our right-handed helices, which are what we predominantly find in proteins in our biological systems, are here. And there are some left-handed helices in proteins, and those will have dihedral angles over here. So if I said earlier, or in the previous video, that amino acids are chiral molecules, and so the chirality is what determines why the dihedral angles here cause a right-handed helix, and the dihedral angles here cause a left-handed helix. If we, instead of the L chirality, the L enantiomers and enantiomers, if our amino acids were instead in the D, the dextrorotatory, then this would actually be the opposite. Having dihedral angles here would give us a left-handed helix, and vice versa for the right-handed helices. But in Earth's biology, our amino acids are almost exclusively L enantiomers, and so we see right-handed helices. So these helices are built like this. You see that this is the right-handed helix going up, and so every one turn, so from here up to here, is 3.6 amino acids. And we see here that, so if this is an amino acid right here, whereas our chain is there, this would be its nitrogen, and over here would be the carbon on its carboxylic acid group. So here is the carbonyl oxygen, and that creates a hydrogen bond with the nitrogen in the amino acid 3.6 amino acids away. So we can see here this is sort of a representation of that, where we have our carboxylic acids creating these hydrogen bonds. So for instance, right here we have this making the hydrogen bond with that, and so that is 1, 2, 3 amino acids away. And so these hydrogen bonds in the backbone of these amino acids is what stabilizes this alpha helix structure. And if we sort of look down into the structure this way, we can look at the way that the amino acids are arranged like this. And so if we start with our, so that was our glycine 245 right here, and that was this one right here. Then the next amino acid in the chain would actually be this one right here, and so we see that we sort of make this kind of pattern if we are looking down into like from the top of the alpha helix. And so you can see that this has the hydrophobic phase here. So we see that we have things like our leucine, and tryptophan and things like that, which form this hydrophobic phase. And so this would be probably facing towards the interior of our protein. So we have our protein sort of globule like this, and this face right here would be facing inward into the protein interior, with this polar face where we have things like arginine and threonine, which arginine are charged molecules, like right here, we have OH, which can form hydrogen bonds, and then we have down here our hydrophobic face. So the polar face is going to be facing out into the water solution, and so that's how these alpha helices can exist in the protein. And I said in the previous video about amino acids, when we have a proline, which, if you remember, has that 5-member ring, it looks like this, back to its own nitrogen. So this would then go to its carbonyl, and so it has these sort of constrained bond angles, which disrupts an alpha helix, as you can see here. And so we have a missing hydrogen bond from it, for instance, and that disrupts the alpha helix, so we don't get this nice straight alpha helix. It causes it to bend, and quite often it will just end the alpha helical structure. And so we find prolines fairly often at one end of the alpha helical structure. And I mentioned before that we have the right-handed helix is the most prominently found in in biochemistry. And the reason for that is we can look at these intracarbonyl steric hindrances. And so that's the carbonyl of one of the amino acids to its own side chain. So right here is probably the best place to see it. So this is like an R, that's a side chain, and this is a carbonyl. And you can see that, well, it's kind of hard to see on this, but it says 3.21 and that's the distance between this carbon here, which would be the the beta carbon on the side chain of our amino acid. And that's about 3.21 angstroms away from this carbonyl oxygen here. So I wrote here that the right-handed is about 3 angstroms range, where with the left-handed it's smaller at about 2.5 angstroms. It's kind of hard to see, but it says 2.42 here. And so if this is left-handed, then we have more steric hindrance between the carbonyl and the side chain inside the same amino acid within the chain. But we also have intercarbonyl steric hindrance. And we can see here that this, for instance, is the carbonyl on the next amino acid over from this side chain here. And if it's once again kind of hard to see, but it says 4.2 something. And so there's about 4 angstrom range between this carbonyl and the side chain in the right-handed helix, where down here you can see this is the carbonyl from the next amino acid over with this side chain of this amino acid, and that's about 2.81. So the left-handed has about 2.5 angstrom range, and so you can see there would be more steric hindrance, which would make these left-handed helices less stable than these right-handed helices, and that's why right-handed is more favored and found more often in biochemistry here on Earth. And so now we'll get to these beta sheets, which these form these sort of nice flat sort of structures here, where we have, so if this kind of kept going and looped around and this kind of kept going and looped around, we'd have parts of the protein chain that create uh, these nice hydrogen bonds here between the nitrogen and the carbonyl on this next chain over. And I showed here that this is going like this. So we have these anti-parallel sheets where you can see, we know that these chains have a directionality to it. So if let's see what the directionality is on here. So this is our side chain here, and this is its nitrogen, and so this would be the N-terminal direction, and this would be the C-terminal. And so our directionality is going this way, but we see in this next one over here that the directionality could be going the other way, but we could also have the directionality going the same way on both, and that would be if we had beta sheet that was like this, and then we sort of had a loop going like this, and then we had a nice beta sheet going like that, so that these would be parallel instead of something like this where it's anti-parallel. So we can see that the sort of hydrogen bonds that we form in the anti-parallel make these sort of nice sort of rectangles where we get more of a, I think this is a trapezoidal sort of hydrogen bonds, where we have this nitrogen-hydrogen forming a hydrogen bond with this carbonyl, so it's somewhat offset from it, where here it's nice and sort of right directly on like this. Uh, and so this is kind of a representation that makes it look nice and flat, but what we actually find in nature is that each of these is going to be slightly offset by about 25 degrees from each other. And so we get this left-handed twist on our beta sheets. And you can see that in this structure here. So beta sheets are, are always shown with this arrow, the arrow showing the directionality of it. So in this case, we'd have parallel beta sheets going. You can see that each one start as we make this loop here. We have this this uh, this twist going on in the beta sheets right here, and as we look directly down onto it, we can see this sort of twist happening pretty good on this beta sheet here for this structure. And then sort of other structures that we need to think about. I mean, the most obvious one is this, which is called intrinsically disordered. And so this structure here, we can see there's this right-handed alpha helix here, which is nice and structured. But then we have this section here where it's 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 intrinsically disordered. There's no structure to it. These parts of the amino acid chain are just kind of flapping around. This, you can see, are we have different structures being superimposed on each other, and so you can see that the different colors, which represent different polypeptides, when we're in this structured area, they overlap real nice, and you can get this nice ordered region here, which will always nicely overlap with other ordered regions. But then when we get to this intrinsically disordered region, there's nothing here. I mean, this shows each color as sort of being in a certain place, but this would actually just be, in solution, it would just be sort of flapping around everywhere. It's intrinsically disordered. So if we want to talk about more structured things, we can talk about, so I said in like the beta sheets, and you'll also see it in alpha helices, there are these parts between the alpha helices and the beta sheets. So you can see here there's alpha helix there and an alpha helix there, and then there's this part connecting them, and this is called a loop. And you can also get it so that the loop is sort of zipped up, so that the structure would actually go all the way up to the end of it. And you see this in beta sheets like this. So this is beta sheet right here, this is beta sheet right here, and then there's 4 amino acids here that form this this hairpin. And you can have different dihedral angles on here. So this is called the I amino acid, which is sort of the first one that's not in this nice beta sheet structure. And then there's the I+1, which can then have the different dihedral angles, and these are the allowed dihedral angles in the hairpin, often called beta hairpin because they're in the beta sheets. And down here is actually from a structure that I obtained in my undergraduate, working on that same beta lactamase enzyme, and this is a hairpin that formed between 2 beta sheets. And so, as I talked about before, the wild type was the OXA 23, And the one I was looking at is the Oxa 239. And so what happened is, uh, so like I said, there was the aspartate to asparagine at the 222 and the proline to serine mutation at 225, which are both found in this region. So in the OX23, what we actually had was more of a loop structure, but then making these two mutations turn it into the nice hairpin structure rather than this slightly more disordered loop-like structure. And so I think before I called it glutamate to glutamine, this should have been aspartate to asparagine. And so it's been a while since I looked at the single letter. So it was D222N. So asparagine is definitely N. Yeah, glutamate is E, so this would be aspartate, yeah. So the way I always try to remember that is glutamate, which has a three-letter code here for glutamate. Its one letter is E, so that spells glue. And yeah, that's just the way that I remember the one letter. So this was aspartate. I think I called it glutamate earlier, but this should have been aspartate. And so it's aspartate to asparagine, which is just essentially that carboxylic acid becoming this instead, becoming this amide here instead. So just making that mutation and that proline to serine, and as I talked about before, proline has that five-membered ring. Oops, that should be, yeah. So C, and so that has that very constrained bond angles, and so that's why we had this loop structure here. And so the loop structure had the proline kind of over here, which kind of prevented this part from making this nice sort of hairpin turn. And we see here that this asparagine, so we can see that the carbonyl oxygen here is able to make hydrogen bonds here, whereas when we looked at the structure when it was in the when it still had the aspartate, then this was sort of off in a different direction, so it was not stabilizing it using these hydrogen bonds. And so we can see that we can turn a loop structure into this beta hairpin by just changing the amino acids, by mutating the amino acids in the sequence here. And so that's sort of these different structures that we find in between the beta sheets and the alpha helices. And so up here, I want to talk about sort of ways that we can look at the different secondary structures, and we do that by doing what's called circular dichroism. And what this does is, we talked about before how we can use polarized light to look into whether something has the L chirality or the D chirality, whether it's levorotatory or dextrorotatory. And so here we can also we can use circularly polarized light. So this is circularly. We have both left and right circularly, or sort of clockwise and counterclockwise together, and it hits our sample and depending on sort of the secondary structure of the sample, then you will create phase shifts in the light, which then will uh, either give you predominantly left or predominantly right circularly polarized light, which you can then detect. And so down here we see that when we shine light onto our protein, then we get these different excitations based on the structure. So our single atoms will have these electron excitations. Then we can also have the peptide bond excitations, which are basically just stretching and compressing these bonds. And same with this shows that as alpha helix. The alpha helix, this structure will actually compress and expand, making these vibrational excitations. And the vibrational excitations is caused by absorbing and then emitting the light, which based on the structure of this alpha helix, it's going to cause phase shifts because it's going to absorb the light, which causes the vibration, and then it's going to emit the light, and it's going to absorb light of one circular polarization more than it's going to absorb light of the other circular polarization. So if this is, say, clockwise, it's going to absorb that. Then our counterclockwise comes in, and that will be less absorbed. And so what ends up happening is once it's emitted, you have this this phase shift, which will then cause this circularly polarized light. So some of it is going to make it through without being absorbed, and so it's not actually going to cause the destructive interference here. It'll cause it between the emitted and the unemitted light of this particular polarization, such that here we have phase like this, where here we had phase like this, and so this causes destructive interference, and so it'll be smaller amplitude, where this, which was not absorbed, will have a higher amplitude. And so then we get this differentiation in the circular polarization based on whether we have these secondary structures. Whereas if this was not in this secondary structure, then we would not have this absorption and readmission going on. And so this would not have that destructive interference, so we would not see any of the change in amplitude. And so we can track these changes in amplitude, and so the different colors on here are at different temperatures, and so you can see, particularly more striking on this one, for instance, we have this line at higher temperatures, where we have this more pronounced line here at lower temperatures, where we have more of the secondary structure, and so we're going to see much more of these amplitudes, these higher amplitudes at when we're in the more structured, the more stable version of this. Whereas as you start getting rid of the structure by increasing the temperature, causing denaturation, then we will not get as much of the circularly of the differential in the circularly polarized light. So we don't get as much of this differentiation. It would look more like our incoming light. And so that's a big way that these structure of a protein like this is sort of studied. We can study we can study the stability and so forth of the protein by looking at, so for instance up here we see that increasing the temperature isn't causing as much of a reduction here, and so this would be more stable than this one, for instance. And so using circular dichroism we can study the secondary structure of protein. And then we want to talk about sort of the changes in conformation of a protein. So now we're looking at conformational changes in protein. And the way that's done is primarily through two different ways. One is such as ligand binding. And so this is our substrate or ligand-free protein here. Then you can see as we bind ligand, you can see that, for instance, this alpha helix is now sort of facing down here, and this alpha helix here is now sort of turned to face more inward here. And so binding this ligand is causing changes in the structure or the conformation of this protein. And you can see here this is saying that our total phi and psi angles, if we add up all the changes in our phi and psi angles between these two proteins here, we see a change in that's greater than 3,400 degrees. So, you know, even if we have just small, like 1 degree and half degree changes in any single amino acid between these two proteins, if we add up all these small changes between all of the amino acids in our protein, then we get then we come out to larger than 3,400 degrees. And so binding to substrates or ligands, as they can be called, can change the conformation of the protein. One other that I didn't get an image of here is you can actually have proteins. So you could have a protein here and a protein here, and then associating them together into a complex might cause some change in the conformation of the individual proteins. And this, as I mentioned earlier, is the quaternary structure of the protein, which is just the association of multiple individual proteins into a single one. And that can also cause a conformational change in the shape, in the structure of the protein. And so binding substrate and binding or associating different proteins can cause these conformational changes. But another big one that we will run into a lot is also these what are called post-transcriptional or post-translational rather modifications, which is something like adding a phosphate group onto it. I talked about in the previous video that our serine, our threonine, and our tyrosine, which all have that OH group on them, we can bind a phosphate onto that, which would look like this, for instance. So we see that this has these two negative charges, and we know from earlier that charge has some effect on the shape of the protein. And so what happens is what we have here, and kinase is just the name of the type of enzyme that add phosphate groups onto these onto any one of these amino acids. So there are what are often called the serine threonine kinases, which will add a phosphate group to serine and threonine. Then there are tyrosine kinases, which will add them to tyrosine residues, tyrosine amino acids. So we see that if we add a phosphate onto here, then it changes the conformation of this polypeptide, because now we can have, for instance, we can have this amino acid here making an ionic bond with the phosphate on here because of the charge on this phosphate. And so adding a phosphate can cause various different conformational changes on our protein. And that this is a heavily utilized way of changing the function of a protein, because the function of the protein is based on its structure. And so say we have this protein here which is not functioning, say because of its shape, it doesn't do what it's supposed to do. And so we can regulate the function of this protein by having an enzyme called a kinase. And there are various different kinases that can regulate particular proteins. So we have a particular kinase comes down, adds a phosphate onto this, and now this protein here becomes functional. And so if we don't want this protein to be doing its function, say our metabolism is in a state where we don't need this protein doing the thing that it does, then we can have what's called a phosphatase, which removes the phosphate, come and turn it back into this. And so this is just waiting around. It's not doing what its function. It's just waiting around for some change in the environment, maybe an increase in sugar because you just ate, or something like that. Some chemical signal signals to this kinase to then add a phosphate, and now this protein, which was just sort of waiting around, it was just sort of biding its time, now becomes the functional version, and now it can be doing the thing that it does. And we'll get more into how this regulation occurs once we get into the biochemistry, because a lot of biochemistry is this sort of regulation. This is a huge part of biochemistry and metabolism and things like that, is this this phosphate sort of way of regulating protein function. And we can look at an example down here. So this is a type of receptor. And so we see we have our transmembrane domains here, which, as I said earlier, this is the membrane. And so this part of the protein here is going to be nonpolar. It's going to be hydrophobic. And this part out here is sticking sort of out of the cell. So this could be outside the cell. And so this part is inside the cell. And so what might happen is some substrate or ligand would come and bind to this, and that might cause a conformational change throughout this, which then allows something like ATP to come into here. And so we can actually phosphorylate it, which then would cause different sorts of conformational changes, which quite often with a receptor like this, when you create a conformational change in it, what that does is it opens up some part of it. It exposes some part of the receptor so that another protein can actually come in and associate with it, and that will cause a cascade of signaling, which once again we'll get into once we get into the biochemistry and metabolism. And that signaling is often done using this right here. So, for instance, some protein might associate with this, which then causes a conformational change in this protein, which then maybe that will allow it to do a phosphorylation of some other protein, so that so we have our kinase, and that will activate this protein, which can then go on and do other things. And so, like I said, we'll get into much more detail about that when we get into metabolism and biochemistry. And so that is everything I think I had for this part here. Uh, yeah, so uh, just sort of a brief review. We talked about the primary structure of the protein, which is just forming these peptide bonds, which, as I said, requires energy. Then we want our protein to fold into its native state. Uh, and so to do that, we use the hydrophobic effect, which keeps the hydrophobic amino acid residues sort of pointing inside, where the hydrophilic are pointing outside. And then we can also use the dielectric effect to uh, to change the pKa and of our amino acids, and generate these ionic bonds. And as we'll see later too, changing the pKa is another way that you can change the function of, say, like an enzyme. If you need a different form of one of these side chains inside your active site, then you can the protein can sort of regulate the dielectric inside the active site of your enzyme. And we'll get into more of that later in a different video. And so we can also look at denaturation of our protein, which is going from our native state to this disordered state, using things like pH and solubility and temperature and ionic strength, which would just be something like adding a whole bunch of salt to it. So you can denature by adding just a bunch of salts to it, or removing salts for that matter. And down here we looked at the denaturation of the protein using temperature, and how just changes in even like a single amino acid can alter the stability of the protein. Then we went over here, we talked about the alpha helices and how they make the right hand twist, and they can have these hydrophobic face on one side of the alpha helix and the polar face on the other side. And we talked about how these steric effects are what stabilize the right-handed helix compared to the left-handed helix, which, if we were using the dextrorotatory rather than the levorotatory, this would actually be the opposite, and these would all be completely mirror reverse from each other. Then we talked about the beta sheets, which we saw are also stabilized by these different hydrogen bonds, depending on if it's anti-parallel and parallel, and it makes this twist that we can see here. And we talked about the different sorts of loops and beta hairpins that we can see between the various beta sheets and between the alpha helices. So we could have this going off, and maybe then it would make another alpha helix down here. And we can once again, we saw that just changing the amino acid, just changing the amino acid sequence. I was, in my undergraduate research, able to change from a loop to this more structured beta hairpin in this beta lactamase from the OXA23 to OXA239. And so that's just sort of more confirmation that the structure of the protein is based on its amino acid sequence. And then we talked about the way we can study the secondary structure through circular dichroism, and there are obviously other sort of more direct ways of actually getting structure, which I'm considering making a whole video on after this. Uh, I'll think about that. But anyway, you could use things like X-ray crystallography or cryo-electron microscopy to actually get the structure. But then you can look at these secondary structures and their stability by using circular dichroism. And then we talked about the way you can cause conformational changes in the protein, which, as I said, was binding these substrates, uh, associations between different proteins. And once again, these things can be related. It could be that binding some substrate sort of exposes part of this protein such that another protein could come and then associate with it, for instance. And another another big way was this phosphate binding, so the kinase phosphatase adding or removing the phosphate from a protein from the serine, threonine, or tyrosine OH groups on those amino acids. And that causes conformational changes. And then we looked at this receptor, that was sort of an instance of this happening. And so given that, I think I will call this video finished. I might add an addendum onto this video just showing a structure using a program called PyMold, just so you can get sort of a quick look at what structure looks like. But this is everything that I believe I'm going to discuss about the structure of proteins, and so I will see you in the next video.